In this video, we can discuss about the sterility testing. Let's start with definition. Sterility testing is defined as the test that critically assess whether a sterilized pharmaceutical product is free from contaminating microorganism. And the sterility testing are intended for detecting the presence of viable microorganism in a test solution or pharmaceutical products. The principle of sterility testing is if a sample removed from a surface or pharmaceutical product is allowed to grow in a culture media at optimum condition, it will produce the turbidity in the medium if it containing any viable contaminating microorganism. Now let's see what are the importance of sterility testing. Sterility testing is an important quality control evaluation test for the pharmaceutical products and equipments which must be sterile while using and which are administered directly into the systemic or blood circulation. So this test is used to evaluate the sterility of pharmaceutical products like parenteral preparation which are injected in different routes like intramuscular, subcutaneous, IV bolus and IV infusions, and implants, then ophthalmic preparations which are directly instilled into the eye, and different equipments which are used for the surgeries like syringes, needle, bandages, surgical dressing and surgical instruments. Now let's see what are the cardinal aspects or what are the vital points to be noted while performing sterility testing. First thing is random sampling of sufficient number of container per batch. It is not practically possible to evaluate the sterility of each containers of a batch. A batch may consist of thousand or lakhs of container per batch. So it is not practically possible to evaluate each container. So we will take the sample in a random manner which will represent the batch. And from the each container we have to take the sufficient portion or volume for the sterility testing which will represent the manufactured sterile product. And the sterility testing should be done under aseptic condition designed to avoid the accidental contamination during the test. And during the sterility testing, the working environment must be adequately monitored at regular intervals by sampling the air and the surface of working area. But during the sterility testing, extremely low level of contamination cannot be detected. The complaints with the test of sterility individually cannot certify absolute assurance of freedom from the microbial contamination, but the total sterility can be assured by strict compliance with good manufacturing practice guidelines of manufacturing products of sterile products. Now let's discuss an outline of total procedure of sterility testing. First, we have to arrange the required sterilized equipments or apparatus in aseptic room where we will perform the sterility testing. Then we will randomly sample specified number of bottle or item from each batch. Then we will measure the specified volume or amount of sample from each bottle or item. Then we will dilute the sample with either fluid A or fluid V if it is necessary to inactivate any antimicrobial agent which are used as a preservative in parenteral preparations. After that, we will select any one method of official method of sterility testing membrane filtration or direct inoculation, and then we will perform this method in laminar airflow unit in order to avoid the contamination of microorganism. After the method, we will transfer the sample or the membrane to culture media which are used officially for sterility testing like fluid thioglycolate media, alternative thioglycolate media and soybean casein digest media. Then the inoculated culture media with the test solution is incubated at 30 to 35 degrees Celsius for bacteria and 20 to 25 degrees Celsius for fungi for 7 to 14 days. Then we will observe for the growth of microorganism in the culture media. So after or during the 7 to 14 days, if there is any growth in the culture media, it can be observed as turbidity. It indicate the failure of test for sterility of the product. And if there is no turbidity, it indicate there is no growth of microorganism. That indicate the pass of test for sterility of the product. As I mentioned before, it is not practically possible to evaluate the test for sterility of each containers of a batch. A batch may vary from one containers to lakhs of containers. So we have to take the sample for sterility testing for a specific number which is selected randomly and which will represent all the batch, and these specific numbers are specified by different official books like pharmacopoeias. So this is an example for injectable preparation, and this is the number of item per batch, and these are the minimum number of item recommended to be tested. Suppose a batch is consists less than 100 containers. We have to take either 10% or 4 container, whichever is greater. Suppose a batch consists of 50 containers. We have to take 10% of the 50 or 4 containers. 10% of the 50 is 5 or 4 container, whichever is greater. 5 is greater than 4, so we have to take 5 containers for sterility testing. If it is 100, we have to take 10% of the 100, that is 10 or 4 containers, so we have to take 10 containers for the sterility testing. So if batch consists more than 100 and less than 500, we have to take 10 containers, and if batch consists greater than 500, we have to take either 2% or 20 container, whichever is less. That means, for example, if a batch consists 600 containers, we have to take 2% of the 600 or 20 containers. 2% of the 600 is 12 or 20 containers, whichever is less. So we have to take 12 containers for the sterility testing. Now coming to the ophthalmic and non-injectable preparation. If batch containing less than 200 containers, we have to take either 5% or 2 container, whichever is greater. If batch contain more than 200 containers, we have to take 10 containers. Coming to the next, for the surgical dressing, if batch consists less than 100 packages, either 10% or 4 package, whichever is greater. And for a batch of 100 to 500 packages, we have to take 10 packages for sterility testing. If batch consists more than 500 packages, either 2% or 20 packages, whichever is less, is taken for sterility testing. Now coming to the bulk solid, if batch consists less than 4 containers, we have to take sample from each batch. If batch consists 4 to 50 containers, we have to take either 20% or 4 container, whichever is greater. If batch consists more than 50 containers, either 2% or 10 container, whichever is greater, should be taken for sampling. So these are the number of containers to be sampled. Now coming to how much volume or how much amount should be taken from each containers. These are the quality of liquid per container versus minimum quality recommended from each containers. So for liquid preparation, These are the quantity in each containers of injectables, and these are the minimum quantity of product that is recommended to test. For example, each container consists less than 1 ml. We have to take total content of the container and 50 ml of the culture media for sterility testing. If each container contains greater than 1 ml and less than 4 ml, we have to take half of the content of the containers and 20 ml of the culture media. If each container consists greater than 4 ml and less than 20 ml, we have to take 2 ml of the sample and 20 ml of the culture media for sterility testing. If each container consists greater than 20 and less than 100, we have to take 10% of the content unless and otherwise specified in the monogram, and 40 ml of culture media. If it is greater than 1 ml per container, not less than half of the content should be taken for sterility testing. Now coming to solid preparation, if each container contains less than 50 milligram, total content to be taken for sterility testing with 40 ml of culture media. If each container consists greater than 50 and less than 200, half of the content should be taken. If each container contains greater than 2 milligram, we have to take 100 milligram from each container and 80 ml of culture media for sterility testing. And for ophthalmic preparation, we have to take 10 to 100 ml and 5 to 10 ml of culture media. And other preparations, the preparation which are soluble in water or appropriate solvent, or insoluble preparation to be suspended or emulsified, 1 to 10 gram with 0.5 to 1 gram of culture media to be taken. So these are the different sampling technique of sterility testing. After taking the sample, sometime it may be required to dilute the sample with either fluid A or fluid B. So fluid A can be prepared by 1 gram of peptic digest of animal tissue or its equivalent in water, and finally we have to made up the volume to 1 liter. Then we have to filter and centrifuge to clarify, then adjust the pH to 7.1 plus or minus 0.2, and then dispense in an 100 ml standard flask and sterilize at 121 degrees Celsius for 20 minutes, or by using fluid B, which is used when the sample containing oil or lecithin as a test solution. Fluid B can be prepared by adding 1 ml of polysorbate to each 1 liter of fluid A. Finally, we have to adjust pH to 7.1 plus or minus 2, and we have to sterilize the fluid at 121 degrees Celsius for 20 minutes. Now coming to the different culture media used for the sterility testing. There are 3 official medias which are used for the sterility testing. First one is fluid thioglycolate media. It is used with clear fluid products. Second one is alternative thioglycolate media, used with turbid and viscid product, and also for the devices having tube with smaller lumina. And third one, soybean casein digest media for fungi. All the media has to be adjust the pH to 7.1 plus or minus 2 and has to be sterilized at 121 degrees Celsius for 20 minutes. And we have to perform sterility testing of culture media also, in order to assure that there is no contamination in the prepared culture media. So this is the composition of different culture media. Fluid thioglycolate media can be prepared by using L-cysteine, dextrose, granular agar, yeast extract, pancreatic digest of casein, and sodium thioglycolate and rosazurin, and to distill water up to 1000 ml, and alternative thioglycolate media with L-cysteine, sodium chloride, dextrose, yeast extract, pancreatic digest of casein, sodium thioglycolate, and finally make up the 1000 ml with water, and soybean casein digest media by using sodium chloride, dibasic potassium phosphate, dextrose, pancreatic digest of casein and papain digest of soya meals, and finally make the volume 2000 ml. Now coming to the different microorganism and incubation time for the different culture media and the organism which are used for control of the sterility testing. Fluid thioglycolate media, we will use a positive controls by giving microorganism like Clostridium sporogenes and Pseudomonas aeruginosa, and incubation temperature of 30 to 35 degrees Celsius, and Clostridium sporogenes is an aerobic microorganism, and Pseudomonas aeruginosa is an aerobic microorganism. And in case of alternative thioglycolate media, we will use Bacteroid vulgatus and Clostridium sporogenus as standard microorganism, and soybean casein digest media, Candida albicans and Aspergillus brasiliensis at 20 to 25 degrees Celsius is used as a standard microorganism for the sterility testing, which is mainly used as a positive control. Now coming to the method of sterility testing. Sterility testing can be either done by membrane filtration method, it is also known as method A, or by direct inoculation method, which is method B. And method A or membrane filtration is used for the sterility testing of oil and oilments, non-bacterial solids which are not readily soluble, and soluble powders, and this is mainly used for the sterility testing of large volume parenterals with volume greater than 100 ml, and direct inoculation method is mainly used for the sterility testing of small volume parenterals. Now coming to the procedure of membrane filtration method, first we have to wash the exterior surface of container with antimicrobial agents. Then we have to transfer aseptically the specified content in laminar airflow unit. Then we have to dilute the testing solution if required with appropriated fluid A or B to inactivate inhibitory action of preservative. Then in case of membrane filtration method, we will filter this solution, test solution, using an hydrophobic edge membrane with a pore size of 0.45 micro. After that, we will wash the membrane with the appropriate fluid A or B to get rid of any possible traces of antibiotics that would have been sticking to the membrane. Then we can either transfer the segregated microorganism or the part of filter paper into two different culture media, that is fluid thioglycolate media or alternative thioglycolate media for bacteria, and soybean casein digest media for fungi, and we will incubate at 30 to 35 degrees Celsius in case of fluid thioglycolate and alternative thioglycolate media, and 20 to 25 degrees Celsius in case of soybean casein digest media. Then we will observe for growth for an incubation period of 7 to 14 days. So sometime the test solution may contain some preservative which will inhibit the growth of microorganism if there any contamination. So that time, in order to inactivate the inhibitory agents, we may add some inactivating agents like for inactivating inhibitory agents like phenol and cresol alcohol by dilution, penicillin by adding penicillinase, parabens by dilution and by adding teen, mercurial compound by adding sulfate compounds, and quaternary ammonium compound by adding lecithin and lubrol lecithin and teen. So these are the different inactivating agents which are used to inactivate the inhibitory actions of inhibitory agents. Now along with the test solution, we may also use some controls, and these controls are used to show that the microorganism will actually grow under the conditions of the test and which are not contaminated. So there are two type of controls. First one is positive controls and negative control. So in positive control, along with the medium, we will add a non-microorganism, and this is used to confirm the culture media is capable of growth of microorganism. So we can use Staphylococcus aureus, Bacillus subtilis, Pseudomonas aeruginosa as aerobic organism, Clostridium sporogenes and Bacteria vulgatus as anaerobic microorganism, and Candida albicans and Aspergillus niger as fungi as a positive control along with the culture media. And we can also use a negative control in which we will not add any sample or any standard microorganism. We will keep incubate a medium without any organism or sample to confirm the medium is sterile. Medium is not contaminated by microorganism. So this is the apparatus used for membrane filtration. It consists of an reservoir and receptacle with an membrane filter with an pore size of 0.45, which is assembled and sterilized prior to use. So this is the reservoir and receptacle, and this is the membrane. Here we are using a membrane filter, and finally after the filtration we will remove this membrane and we will place in culture media and we will incubate for 7 to 14 days. So this is another apparatus which are used in industry for sterility testing. This is an advanced equipment with vacuum in order to increase the speed of filtration. So this membrane filtration can be used for the sterility testing of oily and oily based products, ointments that may not put into the solution, and non-bacteriostatic solid that does not become soluble in culture media rapidly, and a soluble powder or liquid that possesses inherent bacteriostatic or fungostatic activity. Now coming to the second method, that is the direct inoculation method. All the sampling technique is same as that of membrane filtration method, and the procedure is first we have to wash or clean the container, sampled container with antimicrobial agent to assure that there is no contamination of microorganism from the outside. Then we will transfer aseptically the specified content in laminar airflow unit. Then specific volume of sample from each container is to be withdrawn, and dilute the test solution if required to inactivate the inhibitory action of preservative. Then we will directly inoculate this to culture media, that is fluid thioglycolate and alternative thioglycolate, and we will incubate at 30 to 35 degrees Celsius for bacteria, and soybean casein digest media at 20 to 25 degrees Celsius for fungi, and then we will observe the growth of microorganism for 7 to 14 days. So this is by using micropipette we can directly inoculate the test solution into the culture media. That is the direct inoculation method, and this method can be vary according to the substance to be tested, like aqueous solution and suspension, oily and oil solutions, ointments, solids, sterile devices, and transfusion and infusion assemblies. Now coming to the interpretation of result. So after performing the sterility testing method either by membrane filtration or direct inoculation method, the test sample is added to the culture media, and then we will incubate for 7 to 14 days at a respective temperature. So at the regular interval during the incubation period and the completion of 14 days, the media should be examined for the bacterial growth. If a negative evidence is observed in the culture media, it indicates the no growth of microorganism, which will show the sample under the test will passes test for stability. So if the positive evidence is there in culture media, it indicates there is a microbial growth in the culture media. We have to reserve the container which is exhibiting the growth and analyze for the cause of the growth by comparing the growth in the control, positive and negative controls. So we can confirm a test is passed for the sterility testing by observing there is no turbidity, it indicates no growth, and it is giving a negative test in the sterility testing only if the test solution, it containing test solution and medium incubated for 7 to 14 days, is giving negative result. There is no turbidity in test solution, and it is giving a positive result for the positive control, which containing a standard organism and the medium. It indicates the culture media provided for the sterility testing is capable of producing microorganism if we are added a non-standard microorganism, and a negative result for negative control in which we are incubating a culture media alone. It indicates there is no contamination in the culture media, and we can confirm the sample is failed for the test for sterility if the test solution giving a positive result and turbidity, and a positive control also giving a positive result, a turbidity, and a negative result only culture media is also giving a negative result. So during and after the 7 to 14 days of incubation, there are two chances. First one is absence of turbidity, that indicate no growth of microorganism. It indicate negative test for the sterility testing, and the sample will passes the test for sterility. And another chance is the presence of turbidity to the presence of growth of microorganism. It will give a positive evidence. So we have to reserve the containers which are used for the sterility testing in the first test, and we will analyze the growth. During the analysis of the growth, if the sample is found to be unrelated to the sample by comparing with the result of controls, if it is giving a positive result to a negative controls where we had used only culture media for the incubation, it indicates the contamination is from the culture media. This first test is known as invalid. So that we have to perform a first retest with the same number of sample as that of the first test. So after the first retest, after 7 to 14 days, again there are two chances, either absence of turbidity, which indicate no growth of microorganism, there is no contamination, so we can confirm the test sample will passes the test for sterility, or there may be a presence of turbidity, it indicate a positive result. Again we have to analyze the growth of the first retest, and during the analysis of growth, if the sample or microorganism is related to sample and which is not distinguishable from the microorganisms which are present in the first test, sample is considered as failed for their test for sterility. If it is unrelated to the sample and it is distinguishable from the first test, the first retest is known as invalid. So again we have to perform a second retest with the double the sample number of containers, and after 7 to 14 days we can observe either absence of turbidity, which indicate there is no growth, which indicate negative test for sterility, and it will passes the test for sterility. And if there is a presence of turbidity, it indicate a positive result for sterility testing, it failed the test for sterility. So in short, after and during the incubation of sterility testing, if any growth is not observed, it is considered as the sample is passed for the sterility test. If there is a growth is observed, we have to reserve the containers and we have to perform a retest with original sample number as that of the first test. After the 7 to 14 days of first retest, if there is no growth is observed, it will passes the test for sterility. If growth is observed and the growth is not readily distinguishable from the growing those growing in the containers reserved in the first test. It is considered as the preparation or the test sample fail for the test for sterility. If they are readily distinguishable from those growing in the container reserved in the first test, it is considered as invalid. Again, we have to perform a second retest with the twice the number of sample, and after 7 to 14 days, if there is no growth is observed, the preparation will passes the test for sterility. If any growth is observed, the preparation fail in the test for sterility. So that is all about the interpretation of result of sterility testing. These are some references for this topic, and these are some questions from this portion. Sterility testing: write an essay on sterility testing of pharmaceutical product, write about the direct inoculation method of sterility testing, and write a note on membrane filtration method of sterility testing. So that is all about the sterility testing of different pharmaceutical products. Hope it is clear. Thank you for watching this video.